Good morning, everybody. I'm delighted to attend this conference today and to tell you about the localization of neurotrophins, Trk-Receptors, and calcium-binding proteins in mechanosensory systems and retina of notobranchius guenteri. Neurotrophins are growth factors regulating neuronal proliferation, synaptic plasticity, development, survival, growth, and differentiation. Neurotrophins' biological functions are mediated by signals-transducing systems that are initiated by their interaction with the Trk receptors in the central and peripheral nervous systems, including sensory organs. Calcium-binding proteins are also present in these systems. They are involved in essential physiological functions related to calcium ion, such as nerve impulse transmission, neurogenesis, synaptic plasticity, and transmission. Further, calcium-binding proteins are supposed to be involved in neuron protection. Trophins, Trk systems, and calcium-binding proteins are evolutionarily conserved, and they perform their roles in various vertebrates, including fish. However, according to our knowledge, their localization has been never investigated before in the sensory systems of an emergent experimental model, Notobranchius guenteri. Notobranchius guenteri is a small annual killifish affiliated to the cyprinodont clade, adapted to live in temperate savanna pools in Africa and the Neotropy. Living in unhospitable for reptilious habitats made Notobranchius' lifespan extremely fast. Its sensory systems share several morphological and functional similarities with mammals, including humans. It shares an age-dependent organ decline with different vertebrates, and some lesions are also typical of human geriatric phenotype. Hence, it is emerging as a suitable model for translational medicine, and in particular for aging studies. Since some fish sensory organs maintain the capability to regenerate also in adult life, the present investigation aims to provide a morphological description of the retina, inner ear, and lateral eye system of Notobranchius guenteri, and to examine the immunolocalization of neurotrophin Trk systems and calcium-binding proteins to gain insights into their function and potential involvement in regeneration. Hence, adult specimens of Notobranchius guenteri were fixed in wen for 24 hours and routinely processed for histological and histochemical analysis. We carried out immunoperoxidase, single and double immunofluorescence, and fluorescence was detected with confocal laser scanning microscope. So, let's start from the mechanosensory organs. The morphological analysis shown here conformation similar to that one of other actinopterygian ears. The inner ear of Notobranchius guenteri is formed by three semicircular canals, anterior, horizontal, and posterior, the utricule, the saccule, and the lagina. Each semicircular canal has an expanded portion called the ampulla. As shown in these two light micrographs, inside each ampulla there is the sensory epithelium represented by the ampullary crest. In the utricule, saccule, and lagina, the sensory epithelium is organized into macules with slits. Finally, in the photo below, it is possible to observe a branch of the eighth cranial nerve departing from the brain and innervating the saccular macula. In the following slide, I will illustrate the semicircular posterior canal of the inner ear containing the crista ampullaris and the utricular saccular with their nervation. At higher magnification, it is possible to observe the crista ampullaris organization with its supporting cells, hair cells, and the cupola, and the utricular macula with its supporting cells, hair cells, and numerous serous cilia. Notobranchius guenteri lateral line system is made up of three neuromas located both in the head and in the trunk. Here I show you the neuromasses of the head. They have an organization similar to that one of another actinopterygian, the well-known zebrafish. They present central apical sensory hair cells innervated by neuronal peripheral projections. Hair cells are surrounded by two types of sensory cells, supporting and mantle cells. These latter are placed around the supporting cells and complete the mechanosensory organ laterally. In the head, three different shaped neuromasses have been identified. According to their positions, they are called supraorbital, infraorbital, and iomandibular. The neuromasses of the trunk have the same organization and similar shape of the neuromasses of the head. Concerning the immunohistochemical analysis, the immunopositivity to S100 protein allowed to identify cells displaying Trk-A and Trk-C receptors. These two were found in the hair cells of the macula of ventricule, saccule, and lagina. Their pattern of distribution is similar. Both Trk-A and Trk-C were localized in cytoplasm of cylindrical cells placed in the apical part of sensory patches of different macules. These cells were identified as sensory hair cells because of their morphology, localization, and S100 protein immunoreactivity. An immunoreactivity for Trk-A, Trk-C, and S100 protein was also detected in the crescent polaris of the semicircular canals and their innervation. In the same way, confocal immunofluorescence demonstrated the occurrence of Trk-A, Trk-C, and S100 in the central apical portion of the neuromass corresponding to the hair cells. No immunoreaction was found in basal cells, and immunoreactivity through Trk-C and S100 was detected in the lateral line nerve contacting the neuromass 2, and, as you can observe, similar pattern of distribution have been displayed in the different neuromasses with different position in the head. Even if no immunoreaction for Trk-B receptors was found in any structure of the inner ear and lateral eye system of Notobranchus guenteri, the ganglia of the 8th cranial nerve showed an intense and specific immunoreactivity for these receptors. Here, Trk-A and Trk-B has been detected as well. Before to start talking about the localization of neurotrophin Trk system in the retina, I illustrate these hematoxylin eosin light micrographs depicting the morphology of Notobranchus guenteri retina, comprising with 7 layers, its structure a likeness with that of a human counterpart. The immunohistochemical analysis revealed that in the retina of Notobranchus guenteri, retinal pigmented epithelium extensions exhibit immunoreactivity to the receptor Trk-A while lacking immunoreactivity to NGF. In the photoreceptor layer, outer and inner segments of the photoreceptors are NGF immunoreactive, and an NGF Trk-A colocalization occurs in the inner segment. The picture reveals distinct NGF Trk-A immunoreactivity patterns without evident colocalization in the outer plexiform layer. Within the inner nuclear layer, distinct subpopulations of macroin cells were identified as immunopositive for both Trk-A and NGF. Some of them displayed exclusive Trk-A immunoreactivity, while others exhibit NGF Trk-A colocalization. Additionally, certain NGF immunoreactive bipolar cells were observed, and different subpopulations of ganglion cells were NGF and Trk-A immunostained. However, no colocalization views were observed in the ganglion cell layer. Regarding the BDNF Trk-B system, it occurred in the cytoplasmatic prolongation of the retinal pigmented epithelium. Within the photoreceptor layer, the inner segment exhibited concurrent labelling for both BDNF and Trk-B. The outer segment displayed immunostaining for BDNF. The outer plexiform layer demonstrated separate but coexisting immunoreactivity for BDNF and Trk-B. Subsequently, various subpopulations of macroid cells were identified as immunopositive for both BDNF and Trk-B, with instances of colocalization observed. Furthermore, ganglion cell soma within a ganglion cell layer exhibited immunostaining for both BDNF and Trk-B. Concerning the NT3 and Trk-C immunoreactivity, it occurs in the retinal pigmented epithelium cell prolongation. Trk-C immunoreactivity was detected in the outer segment of photoreceptors, while the inner segment exhibited immunopositivity to NT3. Concomitant immunoreactivity to NT3 and Trk-C was identified in the outer plexiform layer. However, the antibodies did not colocalize. Within the inner nuclear layer, bipolar cells displayed NT3 immunoreactivity, and a distinct subset of amacrine cells showed Trk-C immunoreactivity and NT3 immunoreactivity. Furthermore, various ganglion cell subpopulations within the ganglion cell layer were immunostained with NT3 and Trk-C. However, NT3 and Trk-C did not colocalize within the retina of Notobranchus guenteri, allowing to hypothesize the paracrine activity. At another depth level, it is possible to observe that also the outer segment of the photoreceptor layer and bipolar cells were Trk-A immunoreactive. At this level, not only the outer segment of the photoreceptors are immunoreactive to Trk-B, but also the inner segments are. Further, Trk-B positivity occurs in the cellular prolongation inside the inner plexiform layer, in bipolar and horizontal cells. Finally, in the section depicted in the picture, Trk-C immunoreactivity occurred in the inner segment of photoreceptor, in bipolar and horizontal cells. Regarding the calcium binding protein localization in the retina, we found that calretinin occurs in the cytoplasmatic prolongation of the retinal pigmented epithelium and in concert rods of the photoreceptor layer. A subpopulation of amacrine cells in the outer plexiform layer showed immunopositivity to calretinin, and ganglion cells were immunoreactive to this antibody too. Here, I reported a higher magnification of what illustrated above. Parvalbumin was immunolocalized in the cytoplasmatic prolongation of the retinal pigmented epithelium in concert rods of the photoreceptors layer in a subpopulation of amacrine cells in the inner nuclear layer and in the soma of ganglion cells. Here the slide reports pictures depicting higher magnification of amacrine cells and ganglion cells. As 100 immunoreactivity was detected in the cytoplasmatic prolongation of the cells in the retinal pigmented epithelium in a subpopulation of amacrine cells in horizontal, bipolar, and ganglion cells. Finally, also the axon in the inner plexiform layer were S100 immunoreactive. In conclusion, for the first time, the present investigation unveiled the localization neurotrophins, the receptors, and calcium-binding proteins in the inner ear, neuromasses of the lateral line system, and retinal cells of Notobranchus guenteri. This newfound information indicates the influence of these proteins on the biology of Notobranchus guenteri, reinforcing its suitability as a model for aging studies. The implications of these findings could contribute to research on aging-related neurodegeneration within the context of translational medicine. Thank you for your kind attention.